go over the slides and ensure that we haven't left anything behind. So today we're discussing the major themes of anatomy and physiology. We have already discussed the definition of anatomy and physiology, and we said that anatomy is the study of the structure and physiology study is the study of the function. And we also study the connection between anatomy and physiology. Both are inseparable. They are both two different faces of the same coin. We cannot have anatomy without physiology, and we cannot have physiology without the anatomy. So we're going to see how we're gonna utilize our knowledge of the human structure. And anatomy goes down as cell structure, cell anatomy, tissue structure, tissue anatomy, organ structure, organ anatomy, and organ system structure, organ system anatomy, and then the organisms and the gross anatomy. Then there's the physiology. There is the cell physiology, there's tissue physiology, there's organ physiology, organ system physiology, and lastly, there is the gross physiology. So we're going to go into that. Okay, so how do we study the anatomy? So there are multiple different ways to examine the structure of the body. There's inspection, and that is when we visually, we look, we inspect. Is there redness? Is there swelling? Is there bluish discoloration of the skin, indicating there's hypoxia? The patient is suffocating, cannot breathe. Is there yellowish discoloration of the sclera of the eyes, indicating the patient has jaundice? That's an example of inspection. Then there's palpation, when we palpate using our palm, when we feel a structure with our hands. We feel the pulse, we palpate for the pulse. We palpate for the apex beat in the heart. That's when we feel there's the pressure and pulsation, and we palpate to feel a sensation of a structure. Then there's auscultation. Auscultation is listening to the sounds produced by the body. Like when we hear with the stethoscope the lungs, do we have normal lung breathing sounds, or do we have tactile vocal fremitus, or do we hear some sounds that indicate disease? We also auscultate and we listen to the heart sound. Is there a murmur indicating that there's a problem with the heart valves? And we hear the heart sound. Do we hear lub, dub? These are the normal sounds of the heart valves. Or do we hear lub, drup, lub, drup? So that indicates this is a murmur or something wrong with the heart valves. There's dissection, and that is the cutting and the separating of the human body tissues to reveal the tissue relationship. And for that, we use a cadaver, a dead human body. And as we said, back in the day, they used to do this dissection on slaves, living slaves, and prisoners or people who were sentenced for execution. There's also comparative anatomy. In comparative anatomy, it is the study, for example, the dissection of multiple species to learn about their form, function, and evolution. That's very interesting because we found out that the chimpanzees are closer to the humans than the chimpanzees are closer to the gorilla. And that is useful. How could that be useful? How could we take advantage of the monkeys, the chimpanzees, being very close to us in the DNA? We can use that for experiments, for drug experiments. So the medications that we take, when they test them first on the monkeys, we get results closest to that of the human being. Okay? So we use them as animal models for experiment. So if you run the medications on mouse and rats, the results that you're going to get, they will not be as close as they are to the humans, because we are different species. The rats and the humans are different species. And although the chimpanzee and the human being are different species, both of them are very close. So comparative anatomy, we dissect monkeys to see if they are close to us, but they are not 100%. The liver of the monkey looks like four finger projections. There's also exploratory surgery, and that is when we open the living body to see what is wrong. And it is now replaced by medical imaging. Back in the day, if you had abdominal pain, they had to open up your belly to see what's wrong. But now, no need for that. No need to open up your belly to see if something is wrong with the liver, or if there's something is wrong with the intestine. Now, all they need to do is diagnostic. They can take an X-ray. They could do an ultrasound. They could do a CT scan or MRI. Then there is radiology, and that is the branch specializing in is the branch of medicine specializing in imaging. There are sub-disciplines of anatomy, and that includes gross anatomy. Gross anatomy. So gross anatomy is the study of the structures that can be seen with the naked eye. Study of the structures that can be seen with the naked eye. Then there's histology. Histology is microscopic anatomy, and that is we study the structure of the tissues that cannot be seen with the naked eye, and we use the microscope. Sometimes we study finer details that are even smaller or less than microscopic anatomy and histology. We use electron microscope. And there is histopathology, and that is when we study the microscopic examination of tissues for signs of disease. There is cytology. Cytology is the study of structure and function of the cells in fine detail, in ultrastructure, and that may be resolved using an electron microscope. Physiology. Physiology is the study of the function. So physiology uses the methods of experimental science. So there are sub-disciplines of physiology, and they include neurophysiology, and that is the physiology of the nervous system. Then there is endocrinology, and that is the physiology of the hormones. And then there is the pathophysiology, which is the mechanism of disease formation, mechanism of disease, mechanism of action of disease formation. So we will learn about that. We will learn about the neurophysiology and the endocrinology as we go along. Comparative physiology. So comparative physiology is the study of different species to learn about the body function. So we study how are the monkeys different from us, whether or not we can use them as models for scientific experiments. Then the basis for much of our understanding of human physiology and the development of new drugs and medical procedures is based on this comparative physiology. When we compare, maybe we can run this not on a human being, but we can run it on a monkey, for example. Okay, so now we're going to talk about the origins of biomedical sciences. We alluded to that a little bit in the past. We said that science today is obviously not what it used to be back in the day. Hippocrates. Hippocrates was a Greek physician. He's known as the father of medicine. He established a code of ethics known as the Hippocratic Oath. So the doctors take that oath before they embark on their profession. That urged physicians to seek natural causes of disease rather than attributing them to acts of the gods and the demons. Because back in the day, people think you are ill and sick because you are possessed by the devil and because you are the demon. And people have really wrong and terrible understanding of science. I mean, there was a woman in South Africa. She was killed because she was accused of cheating on her husband because the baby was albino. You know, the albino children. Yeah, so they said, okay, you cheated on me with a white person. She said I did it, and then they actually killed her. So that shows you people's lack of knowledge in the science can lead to very dangerous things. And then and then Hippocrates is the one who urged people to develop science and to start attributing disease to the natural causes and to scientific things rather than being sick because of the devil or demons and all of that, which may not be entirely wrong. We definitely believe that there are causes of disease that are not testable by science. I believe in that. Some aspects of the pathogenesis can sometimes not be attributed to science, but in the end, we only believe and we only examine and we only discuss the stuff that is testable, the testable hypothesis, the falsifiable hypothesis, and everything else we leave it. We cannot put science into things that are not testable, even if they may have an effect. Even if you believe in demons, even if you believe in devil, even if you believe they can cause a sickness or disease, nobody's telling you that that's not the right belief. It's up to you. But we're telling you that that could not be placed in science and cannot be run into in the scientific method in an experimental design. And then there is Aristotle, who believed that diseases had supernatural and physical causes. So these are called supernatural causes of disease theology. So he did believe that there are aspects of disease that cannot be attributed to science. And he believed complex structures were built from simpler ones. So now we're looking at the development of science over the years. And then came Metrodora. Metrodora was a Greek physician who was a female. She was the first woman to publish a medical textbook. What do you think that medical textbook was about? It was obstetrics and gynecology. So um, she published a book discussing the diseases that involve obstetrics and gynecology. Then came Claudius Galen. Claudius Galen was a physician of the Roman gladiators. So when the Roman gladiators fight each other, he would come and he would heal their wounds, and he would take sample of their blood and sample of the pus and investigate that. And he did come up with very important contributions. Many of them were wrong, but some of them were true and hold true up to today. He did animal dissections because the use of cadavers at the time was banned. And he saw science as a method of discovery. And the teachings were adapted as the dogma in Europe and the Middle Ages. His teachings back in the day were, the most important thing of their time. Okay, then Maimonides came, and this was a Jewish physician. He wrote 10 influential medical texts, and he was the physician to the emperor, the Egyptian sultan at the time, Saladin, which happens to be the name of the book. Yeah, very interesting. Okay, then there's Avicenna, and he wasn't a Jewish physician. He was a Muslim physician, and this was during the Muslim Golden Age, and he combined both Galen and Aristotle's findings with original discoveries. He wrote a book called The Canon of Medicine, but canon means law, the law of medicine, which was very interesting because people think law and medicine don't go together. There's law school and there is medical school, but he combined them together. You know, in the law, they say if you commit a particular crime, then they negative feedback, right? Why do they send the criminal to jail? So he can stop doing to negative feedback. So when he come out of jail, he does not do crime. So Avicenna was telling the same thing, that the law and medicine are very similar. If you commit this crime, you get that particular sentencing or that particular verdict. And in medicine, if you are sick, you get that particular medication. So they are. He was pointing to the similarity between medicine and law. Okay. So this just shows us that, medical art has changed. So this was back in the day how they used to think the human body looked like, and it developed over the years. Then modern medicine started to rise, and that is the beginning of the modern Western medicine was with Andreas Vesalius. So Andreas Vesalius, at the time, the Catholic Church relaxed the restrictions on the dissection of the cadavers, and he performed his own dissections rather than having the barber surgeons. So what does that mean? Back in the day, the barber surgeons, because they didn't know embalming, so the cadaver used to stink and smell bad, and it was putrefied. So nobody can dissect it. But still, they used to dissect it like that. So they had a job for somebody known as the barber surgeon. So the barber surgeon would stand next to the rotten, putrefying, stinking cadaver and run the dissection, while the scientists would sit at the top. But Andreas Vesalius, the place that the scientists used to sit at the top, is known as the cathedral. So Andreas Vesalius was the first one who came down from the cathedral, and he did the actual digestions on that putrefying uh, cadaver, and he published the first atlas of anatomy known as De Humani Corporis Fabrica, the structure of the human body. So it's very interesting for you to remember that the first atlas of the anatomy was published by Andreas Vesalius. Then there is William Harvey. So William Harvey is one of the earliest physiologists, and his contributions represent the birth the birth of experimental physiology. He published the book The Motion of the Heart, and he talked about very important things in the structure and function of the blood vessels. We will talk more about that. Then microscopy extended the ability to visualize the life at the cellular level. So we have Galileo, and Galileo was a physicist who was concerned with telescopes. So in a telescope, they had an ocular lens that is close to your eye. That's why they call it ocular lens. And then a second lens that is very far and close to the object you want to see, known as the objective lens. Then Galileo patented the compound microscope as a byproduct of his work with telescopes, and but he did not use it for studying biological material. But he helped, in the making of this um, um, the ability to visualize smaller objects using two lenses. What was the name of the lens closer to the eye called? Ocular lens. Very good. And the lens that is very far near the object you want to see is known as the objective lens. Very good. Then Marcello Malpighi was the first to use the compound microscope to study the biological material. Observe the blood vessels, capillaries, and capillary blood flow. Then Robert Hooke came in, and Robert Hooke made many improvements to the compound microscope, and he invented the specimen stage. So he developed the microscope and the illuminator and coarse and fine focus controls. His microscopes magnified only 30 times. And he was, so Robert Hooke was the first to see and name the cells. He published the first comprehensive book of microscopy, known as Micrographia, in 1665. So this is Hooke's microscope. Oh, very interesting. Okay, then Anthony van Leeuwenhoek, and he was the one who really took the microscope to the next level in the birth of the modern medicine. So he invented a simple single lens, microscope with great magnification to look at fabrics, 200 times magnification. Then there was a superior magnification. It was superior magnification compared to Hooke's microscope due to Leeuwenhoek's superior lens making. He published his observations of the blood, lake water, sperm, the bacteria from both scrapings and many other things. So it was Anthony van Leeuwenhoek who was able to invent the simple single lens microscope with magnification that goes up to 200. Because remember, the one before him, Robert Hooke, can only see 30 times the magnification. But Anthony van Leeuwenhoek gave us magnification of 200 times. And there was superior magnification compared to Hooke's microscope due to the superior lens making. So he invented a single lens microscope with a better magnification. Okay. Then the cell theory began with Matthias Schleiden and Theodor Schwann. And they examined the wide variety of specimens, and they concluded that all organisms were composed of cells. And we talked about that. So all organisms are made up of cells, and all the cells are made up of tissues, and all the tissues give us organs, and all the organs give us an organ system, and the organ system give us the organism. And we began today by saying that all the cells have different function, they have a different physiology, and therefore, in order to do their job, to do their function, to do their physiology, they must have what they must have. What do they must have? If they have a certain function and a certain physiology, then in order to be able to do that physiology and to do that function, then they need to have the special structures, specialized structures and specialized anatomy to do that job. And we know that cells only come from other cells by cell division. Cells don't come out of nowhere. So cells come from older cells. Same thing, organisms come from other organisms. So we come from our parents. Our parents come from our grandparents. Our grandchildren came from our children, and so on. Same thing with the cell. Cells come from older cells, from other cells that preceded them. All functions of the body are interpreted as effects of the cellular activity. Okay, so the modern biochemical science advances. So we have the germ theory of the disease, that these germs, the bacteria, the viruses, and the fungi and the parasites, they are able to damage our cells using toxin, endotoxins, exotoxins, reactive oxidative species, destroying with the degradative enzymes, metalloproteinases, degradative enzymes, that lysosomes that can destroy the structural integrity of our cells. Then there were advancements in the field of the mechanisms of heredity. So now we know that there are autosomal diseases and there are diseases that are inherited in the sex lineage, known as sex-linked diseases, and we will talk more about them. The structure of the DNA was discovered, that is double helix, and we talked earlier about the central dogma of biology, that we have nucleic acids. We transcribe the nucleic acids of the DNA to get nucleic acids of mRNA, and then we translate the nucleic acid of mRNA into the structural and functional proteins that we need to survive. Disease imaging and treatment. So now we are able to use diagnostics, and we are able to use we are able to use diagnostics, and we are able to use CT scan and X-ray and ultrasound to help us see the processes even better than our own naked eye. So back in the day, they used to open up the abdomen to see what's wrong. Now you don't have to open it, and the X-ray will give you a better, finer detail with the mRNA and with the CT scan, even than you looking with your own eyes and with the microscope. So that's a huge advancement. Okay, then we came to the scientific method. So the scientific method now we're going to talk about the inductive method, known as the scientific method. We're going to talk about the scientific method. So the scientific method is the process of performing science, including careful observation, logical thinking, and proper analysis of the observations and making conclusions. So science and scientific methods set standards for truth. So now let's learn more about that. There's the inductive method. So the inductive method is a process of making numerous observations until one becomes confident in drawing generalizations and predictions. So the knowledge of the anatomy is obtained by this method. So what is proof in science? In science, there are reliable observations that are repeatedly confirmed, not falsified by any credible observation. So that shows the process of making numerous general observations until the one becomes confident in drawing predictions and generalizations. But these, we need to have reliable observations that are repeatedly confirmed, and they are not falsified by any credible observation. In science, all truth is tentative. In science, all truth is tentative, and proof beyond a reason is we need to prove beyond a reasonable doubt, and they may abandon yesterday. We should abandon yesterday's truth if it is negated by today's or disapproved by today's scientific evidence. Then there is another model known as the hypothetical deductive method, and most physiological knowledge is gained by this method. So the investigator formulates a hypothesis. And uneducated speculation. So the hypothesis is an uneducated speculation or a possible answer to the question. So the good hypotheses are consistent with what is already known and testable. So now we need to learn what is falsifiability. So falsifiability means that if we claim something is scientifically true, then we must be able to specify what evidence it would take to prove it wrong. So what I believe is true, I should be able to tell what evidence it may take to prove that it is wrong. So for example, let's say somebody is claiming that the medication hydrochlorothiazide, which is a diuretic, is supposed to drop blood pressure by decreasing blood volume. So in order for me to believe that this is true, I must be able to specify that if you take a pill of hydrochlorothiazide and your blood pressure does not drop, then that means hydrochlorothiazide is not a blood pressure medication. So now I did a good job outlining what should be done to prove me wrong. I already proved that hydrochlorothiazide is a blood pressure medication, but I also need to prove what should be done to prove it wrong. So also, if I believe that Tylenol brings down pain, how can I falsify that? Let's say you guys discovered, you made in lab Tylenol, and Tylenol is analgesic. It shuts down nociception. It shuts down it's an NSAID that blocks the enzymes cyclooxygenases and stops the sensation of pain. Now, how can you guys falsify that? Can somebody tell me how to falsify that? Yeah, so what do we need to say? So if I'm saying that our medication, we have invented Tylenol, and Tylenol brings pain down, pain sensation down. If you take Tylenol, you you will not feel pain. How can I falsify that? Yeah, excellent. So what did you say? Yeah, I heard you. You're right. I just want to see, what did you say? Yeah, what did you say? Okay, so if you give a placebo, that's a good. Yeah, so so then that's not placebo. If you tell them this is Tylenol and it is not, then this is not placebo. Placebo is, it is placebo, but they don't know. They never know. But what your friend said, she really got it. She really got the concept. So to falsify something, as your friend said, I must say two things. Number one, you take Tylenol, pain will go down. And I'm going to falsify that. If you take Tylenol and your pain does not go down, then you have falsified it. Yeah, now we're talking business. So we need both. We need to have a hypothesis that is falsifiable. Okay? Call Domino's. If you don't get your food within 30 minutes, it's free. Now they're falsifying things really well. Okay? So we need to be able to falsify. Then we should have an experimental design. So the experimental design must have elements. So there is a sample size. The sample size is the number of subjects in a study. Number of subjects in a study. And there are controls. These controls are control group. They resemble the treatment group, but does not receive the treatment. So here, they are a control group. So you bring 100 people. You say, we have a pain medication. We have a pain medication. We call it Tylenol. We need 100 people who have pain, who have joint pain. You divide them into 250 groups. 50 people, you give them Tylenol. The other people, you give them a tab with water or sugar. That's the control group. But you don't tell them, listen, this is Tylenol and it is not Tylenol. And you don't tell them this is placebo. You just give it to them. Actually, yes, you do tell them. You do tell them that. You do tell them that this is Tylenol, but it really isn't. You're right. You're right. You tell them that this is Tylenol, but it isn't. Okay? But the problem is when you tell them this is not Tylenol and then it is Tylenol, then you have interfered with the experiment. So I misunderstood you earlier. So that's the placebo. So there's also experimental bias. Experimental bias is associated with double-blind method, is avoided with double-blind method. So experimental bias is when we have our own cognitive bias getting in the way of us getting results. So the experimenter can sometimes be so eager, over eager, that he or she, the experimenters, end up interfering with the results. So what does double blind mean? Double blind means that neither the subject or the experimenter know if the subject is part of the control or part of the treatment group. There is statistical testing. Statistical testing is very important. So you must remember that. How do we repair experimental bias? How do we ensure that the experiment that we did does not have bias? How do we avoid that? And the person who's doing the test, what? Shouldn't know. So we call that double blind. We call that double blind. So I'm running the experiment. I don't know which group is control, which group is placebo. And the participants, the one in placebo, they don't know if they are placebo or control, or they are placebo or treatment. And the other group doesn't know if they are placebo or treatment. Why is statistical testing important? It's important because it provides statement of the probability that the treatment was effective. So for example, if I take Tylenol and my pain goes down by 2%, is that statistically significant? It just went down by 2%. But there's another medication, if you take it, the pain goes down by 90%. So first one, I was feeling pain 10 out of 10. 10 is the most pain and 1 is the least. 10 is the most pain and 1 is the least. I take the medication, now my pain is 9.8. So it's not 10, but it's 9.8. Is that medication statistically significant in helping us? No. Then another medication that I take, my pain was 10, and now it's 1. So we drop down by 9. Is that statistically significant? Yeah, so that's why we need statistics. Peer review. Peer review is a critical evaluation by other experts in the field, and that is done prior to funding or publication. It is done by using verification and repeatability of results, and it ensures honesty, objectivity, and quality in science. So we take the counsel of other scientists, and how can they contribute into the improvement of my experimental design? Maybe they can tell me, while you're doing this experiment, you can do this also. Hit 2 birds with 1 stone. While you are doing while you're doing this particular experiment, the stuff that you're doing has already been tested before. You should go and read the paper and see if you can develop your experiment into something that is more effective and more contributing to the science world. Okay, facts, laws, and theories. So they express the understanding of nature with facts, laws, and theories. So scientific fact is the information that can be independently verified. And the law of nature is a generalization about, so what's a fact? A fact is a piece of information that can be verified, okay? If you say, my phone number is 1234567. It's a fact if I call 1234567 and you pick up. That's a fact because I verified that is true. Then the law of nature. The law of nature is a generalization about the way matter and energy behave. So the results from inductive reasoning and repeated observations, it is written as verbal statement or mathematical formula. Theory. So the theory is an explanatory statement or set of statements derived from facts, laws, and confirmed hypotheses. It summarizes what we know and suggests directions of further study. So that's the theory. It is an explanatory statement that explains or sets statements derived from facts, laws, and confirmed hypotheses. So the law of nature is a generalization about the way the matter and the energy behave. Okay, so what is natural selection? That's a very important thing to understand. So the theory of the natural selection is an explanation of how the species originate and how they change through time. They help understand the human body. They help us understand the human body. And so we are learning about now the population, the species. How do they change? So now we're not studying homeostasis. Now we're studying the stability, not of the internal environment, but the stability of the entire population. So Charles Darwin is the pioneer and the influential biologist of this field. He presented first a well-supported theory of how the evolution works. So on the origin of species, his book is known as the origin of on the origin of species by means of natural selection. And then he wrote another book known as The Descent of Man, which is human evolution and relationship to other animals. So what is evolution? Evolution is change in genetic composition of population of organisms. So that means the development of bacteria resistance to antibiotics. So if you give one antibiotic, penicillin or vancomycin, to somebody who has a bacterial infection, so do you think 100% of the bacteria are going to die? No, about maybe 98%. But that 98% is gone, 2% stay behind. That 2% stay behind are going to now start to increase in size. When they increase in size, if you give them penicillin again, you think now they're going to die? They're not going to die. Only about maybe 30% of them or 40% will die. So now they have developed resistance due to evolution. Then there is natural selection. Natural selection explains how evolution works. So evolution takes place by the vehicle. The vehicle that makes evolution happen is natural selection. So natural selection is how evolution works. So selection pressures, selection pressures are the forces that promote reproductive success of some individuals more than others. And for example, the predators. So predators who are more fit, who are really strong, and they are able to they are able to survive and hunt the prey better than other predators. They will survive. The other predators are gone. Okay, adaptations. So adaptations are inherited features of anatomy and physiology that evolved in response to pressures and that enabled the organism to succeed. So if you better camouflage, so if the lizard can better camouflage to the jungle, then that will make it hide better. It will hide excellently better than other to protect itself from other predators. So some human anatomical and physiological features can be traced back to ancestral primates. Okay, so this is really important. It's really important because some human anatomical and physiological features. So let's talk about our basic primate adaptations. So the early primates were arboreal. What does arboreal mean? Arboreal means they live on the top of the tree, and they have mobile shoulders. And these mobile shoulders have better movement among the branches, and and then we evolved opposable thumbs, and prehensile hands, enabled to grasp the branches and manipulate the objects. And then we develop forward-facing eyes. So our eyes are not like the fish, they are on the side. Human beings are the only ones who have their eyes really in the center. We don't have peripheral eyes, we have central eyes, and that has evolved to help us survive. When you have both eyes in front, you can see predators, you can see harmful, dangerous things, and it helps you also to see the good things you need, like the fruits, the vegetables, and the prey that you need to catch and hunt to eat it and survive. So that helped us with stereoscopic vision, with depth perception, and there's color vision where we find we can find ripe fruit, and then we developed we develop color vision. We can see the color really well, so we can tell which fruit is ripe, which one is not. And then there is large brain, and the large brain is memory, allowing efficient food finding. So we can remember where the food was, and we can start putting information together to help us get to our goal. Okay. So now we have other human adaptations that are shared with other primates, which is walking upright. That is known as bipedalism. So we're not the only ones who walk on 2 feet. So standing and walking on 2 legs is known as bipedalism. What does bipedalism mean? Yes, standing up and walking on 2 legs. And that helps us spot the predators and carry the food and the tools and carry our infants. Okay, and adaptations to the living. These are adaptations to living on the savanna, that is the grassland, as Africa became hotter and drier. So there are skeletal and muscular modifications, and there are changes to the structure. So we have adaptations to living on the savanna, to adaptation of living on the savanna as Africa became hotter and drier, and the skeletal and muscular modification and changes to family structure. Then walking upright. So bipedalism has evolved now. So Australopithecus was a bipedal primate genus that lived more than 3 million years ago. So they were bipedal. Then Homo genus appeared about 2.5 million years ago, which means about 500,000 years after the Australopithecus. And what happened is they were taller and they had larger brain volume, and they were able to make tools. They can sharpen their stone and use it as a knife to cut things. There's Homo erectus. So Homo erectus appeared 1.8 million years ago, and it migrated from Africa to parts of Asia. So the Homo sapiens originated in Africa about 200,000 years ago. And evolutionary medicine traces some of our diseases to differences between modern and prehistoric environment. The hierarchy of complexity. So we talked about that. Who's at the base? The base is the molecules and the atoms. On top of the molecules and atoms, who do we have? Organelles, composed of molecules. On top of the organelles, composed of molecules, we have cells, composed partly of organelles. Then the cells composed partly of organelles are tissues composed of cells. On top of the tissues, we have organs composed of tissues. Then we have organ systems composed of organs. And then we have organism composed of organ systems. And then we have human organization based on successive levels of hierarchy. Okay, body structure and hierarchy. So here you see all the way in the bottom at the base, we have the atom. Many atoms give us a molecule. The molecules give us macromolecules. Macromolecules give us an organelle. Organelles give us the cell. Cells give us a tissue. Tissue give us the organs. Organs together give us organ systems that are working together. And then organ systems give us the organism. And many organisms give us the population. Who can give me an example of an organ system that I mentioned earlier? The one I mentioned when we began class. An organ system. I mentioned it earlier. I said that the skin, yeah, the integumentary system, it does not only have one type of tissue, does not only have one type of organ, doesn't just have epithelial cells or dermal cell or epidermis. It has what else? It has nerves. Remember we said that it has nerves. It has blood vessels. It has hair follicles. So all of these are different organs that are working together like an organ system. The hierarchy of human complexity. So the organism is a single, complete individual. And there's an organ system. The organ system is a group of organs with a unique collective function. For example, the circulation, the respiration, and the digestion. So the organ is a structure composed of two or more tissue types that work together to carry out a function. So an organ has defined anatomical boundaries that can have organs within organs. Tissue. So tissue is a tissue, similar cells and cell products forming a discrete region of an organ and performs a specific function. So that's the tissue. Tissue is made up of the cells. So we have similar cells and cell products forming a discrete region of an organ and that performs a specific function. Cells. Cells are the smallest unit to carry out all basic functions of life. Then there are organelles. Organelles are structures within a cell that carry out a function. And then we have a molecule. Molecules are particles composed of two or more atoms, and the largest molecules, proteins, fats, DNA, are called macromolecules. And atoms are the smallest particles with unique chemical identity. Okay, the hierarchy of complexity. There are two complementary approaches to studying and understanding human life. There is reductionism and holism. Reductionism is a theory that large complex systems can be understood by studying their simpler parts. We are parts that are made up of smaller parts. We are larger parts that are made up from smaller parts. And that is essential to the scientific thinking. But some properties cannot be predicted from individual parts. Holism. Holism is emergent properties, and they occur as we ascend the levels of organization. These cannot be predicted from the properties of individual parts alone. Humans are more than the sum of their parts. So no two humans are exactly alike. So there is always variation. So when we study anatomy and physiology, everything we study, does that apply to 100% of the population? Does everybody have a heart like the heart we're going to study? Does everybody have a lung like the lung we studied? No. There's some people, like this shows you that some individuals lack some muscle. They don't even have the muscle. Some individuals have a typical number of vertebra. They have more bones. Some people have a typical number of organs, and the structure of the organ looks like a horseshoe, or maybe they have a kidney that is in the pelvis, not up there where it belongs. And some people have situs inversus. So situs inversus is very important because there is a left-right reversal of organ placement. So yeah, you've seen that. You see? Yeah, but the heart was on the right side. Yeah, the heart was on the right side. Yeah, you see, some people are like that. So that shows you there is anatomical variation. So this is what we study in anatomy and physiology is for the most part. Most of the people are like that. But it is definitely not 100%. Some people have variations. Oh, there we go. So that shows you that the kidneys normally is supposed to look like that. But some people have a pelvic kidney, a kidney down in the pelvis. Some people have a horseshoe kidney. Then this is a normal heart. But some people have hearts with their aorta that branch like this. Look, you see, their aorta does not look like the normal one. So that shows you there are variations in the branches of the aorta.
And it's like that. Does it impair the functioning of the heart?
It does not impair the functioning of the heart in a statistically significant clinical problem. It's just a small variation, but it's not a really big problem. But in some cases, it can develop into a congenital disease. It can develop into a problem that is really a big problem. But it can be a benign congenital malformation, and sometimes it could be like a really dangerous congenital malformation, failure of embryological development to happen in the normal way. Sometimes these variations, they're not always bad. Sometimes they're positive. They discovered that some people, for example, if you have sickle cell anemia, you will never get malaria. Some people also, when they are born, their white blood cells are lacking a side receptor. This side receptor that is found on all white blood cells. And that is what the HIV uses in order to put its mRNA into our white blood cell. It used two receptors, a main receptor and a side receptor. Some people have a mutation, that side receptor is not there. So do you think HIV can put its mRNA into their cells? No. So that means they will never get HIV. They'll never get AIDS. So that shows you sometimes mutations can actually be helpful. But for the vast majority of times, they are harmful. Okay, so now we're going to define negative feedback. We did that. Then we're going to define positive feedback, and we did that. And then we're going to define gradient, and we did all of that. So let's look at the characteristics of life. So life is a collection of properties that distinguish the living from the non-living things. So life is a collection of properties that distinguish the living from the non-living things. So there are characteristics. We can never define life with a clear-cut definition. But we can observe and notice that if you are alive, you must have the following characteristics. They are over there. Number one, what is the first characteristic? Organization. So living things exhibit a higher level of organization than non-living things. Number two, cellular composition. So living matter is always compartmentalized into one or more cells. Compartmentalized into one or more cells. Then metabolism. Metabolism is the sum of all the internal chemical change. So can the dead be able to perform metabolism? No, they cannot. Then responsiveness, excitability, reflexes, restoring homeostasis. So the ability to sensor. A sensor is number one in the stage of events that we use as a negative feedback to restore homeostasis. Who knows the answer? A sensor is number, I'm sorry? I'm asking the sensor. You see, what does the responsiveness say? Responsiveness says excitability. Excitability is the ability to sense and react to changes in the environment, stimuli. So when we sense a stimuli, what is the number of in the sequence of events of negative feedback that help restore homeostasis? Is the first one. So we must have the ability to sense a deviation so we can respond to it. Then there's movement. Movement is the movement of the entire organism or of substances within the organism. There's more. Homeostasis. The living are able to maintain homeostasis. So maintaining relatively stable internal conditions. So no matter what deviations are there, we're able to restore homeostasis. Then there is development. So development is the change in the form or function. Change in the form or function over time. So we have differentiation, and differentiation is the transformation of unspecialized cells into cells with a committed task. A cell with a committed task. And there's growth. So growth is an increase in the size, and it occurs through a chemical change. There's reproduction. In reproduction, organisms produce copies of themselves and pass genes to their offspring. And finally, we must be able to, as living things, we must be able to have to propagate evolution. So evolution is the genetic change from generation to generation, and it occurs due to mutations. Mutations are change in DNA structure. So we observe evolution in a population as a whole, and a single organism does not evolve over the course of its life. Now we finished with anatomical variation. Now we're going to move into physiological variation. So physiology is even more variable than anatomy. So there are variations in sex, age, diet, weight, physical activity, genetics, and environment. So what does that mean? That means, let's say a child who is 6 years old, pediatric, has bronchitis, then, or they have a lung infection, they have pneumonia, a baby, then a young man and a young woman, they have also pneumonia, infection of the lung. Then someone who is 90 years old have pneumonia, infection of the lung. Are we going to give them the same medication with the same dose? So that shows you we must account for that variation. Variation in the sex, male versus female, variation in the age, pediatric versus geriatric, and then the weight, obese patients versus malnutrition patients, and then physical activity, genetics, and the environment. There are typical physiological values. So when we are talking in anatomy and physiology, we mainly talk about a reference man and a reference woman. The reference man is 22 years old, 154 pounds, light physical activity, and consumes about 2,800 kilocalories a day. While the reference woman, same as the man, except that she's 128 pounds and 2,000 kilocalories a day she burns. Failure to consider variation can lead to over-medication of the elderly and or medicating women on the basis of research that is done on men. Okay, so we need to take into account these different variations. Negative feedback and homeostasis. So homeostasis is the ability to detect change, sense, first step, detect change, sense, activate mechanisms, meaning input signal, input sensory signal, and then integration, then an output signal that oppose it, negative feedback, and then thereby to maintain a relatively stable internal environment. There are two scientists we need to know really well, and that is Claude Bernard. Claude Bernard noted fairly constant internal conditions despite changing external conditions. For example, temperature. And then there's Walter Cannon. So Walter Cannon coined the term homeostasis. Homeostasis. So negative feedback allows for dynamic equilibrium with a limited range around a set point. So Walter Cannon is the one who coined homeostasis, and the body senses a change and negates or reverses it. There is a loss of homeostatic control that causes illness or death. Negative feedback and homeostasis. Homeostasis is internal conditions. In homeostasis, internal conditions fluctuate within a limited range. There's dynamic equilibrium around a set point. There's negative feedback. So in negative feedback is a mechanism that keeps a variable close to set point, and the body senses a change and reverses it. Because feedback mechanisms alter the original changes that triggered them, they are called feedback loops. So for example, homeostasis in the body temperature. So if too warm, the skin blood vessels dilate, causing vasodilation, and sweating begins, where there's a heat losing mechanism. If too cold, the skin blood vessels constrict, and with vasoconstriction and shivering will begin. We talked about that. We talked about all of that. So here the book is really nice because it gives you it gives you an example beginning from very far. So in your house, if the room temperature falls to 19 degrees Celsius, what is that an example of? Hyperthermia or hypothermia? Hypothermia. So the thermostat detects that, and after it detects it, it integrates and sends an output to the effector. Who's the effector here? Who's the effector here? Who's the effector? Who's the effector? Yeah, from looking at the picture. We know there's a sensor and there's an integrator and an effector. Who's the effector? What does the effector do? What does the effector do? The effector mediates the effect. So we know that when blood vessels dilated, they are the ones who responded to the integration. So who integrated here? So when you integrate, what do you do when you integrate? When you integrate, you realize there is either an increase or a decrease, and you need to repair it by negative feedback. And then you send the actual output of repair. The actual output of negative feedback repair is done by the effector. The effector is the one who fixes, who does the action that repairs homeostasis. So who here is our effector? Yeah, we're looking at that picture. Yes. So don't say this. Read it and tell me what it's called. Yes, read the whole cycle. Read the whole cycle, 1 through 6. Take your time. Don't answer me now. Take your time. Read the whole steps. Number 1, room temperature falls to 19 degrees Celsius. Thermostat activates the furnace. Furnace makes heat output. Room temperature rises. Thermostat shuts off the furnace. The room cools down. And then we stop. Who here is the effector? The furnace. So the furnace is the one who did the heat, the one who did the actual thing. But who's the integrator? The thermostat. Who is the sensor? The room temperature is the stress, is the hyper. It went up. So that's what we sense. That's the hyperthermia. So that's not the sensor. We sense the hyperthermia. Who sensed that there's a hyperthermia? Who sensed that there's a hypothermia? I can't hear you. The thermostat. The thermostat sensed there is a hyperthermia that needs to be repaired. Then who is the integrator? The thermostat. Because who told the furnace to become hot? The thermostat. So the thermostat is 2-in-1. Remember I told you earlier, you can have 2-in-1. Remember I told you that the pancreas was the integrator and an effector at the same time. It integrated, there's an increase in glucose and it sent out insulin. So sometimes you can have 2-in-1. Okay, if now the temperature is too hot and the thermostat now is now realizing there is a fall in temperature, there's not an increase in temperature, there's a drop in temperature. Now it's too cold. So then it turns on the AC. Who's the integrator? The thermostat. And who's the effector? The AC, because the AC is the one who did the effect of repairing the homeostasis. Okay, so this is now what's happening in our thermogenesis. As time goes on, if the temperature goes up, you see that's the temperature. That's the U. That's euthermia, 37 degrees Celsius. Do you see euthermia? 37 degrees Celsius, right here. Temperature went up, heading up to 37.5. Is that hyper or hypothermia? Hyperthermia. So now that's hyperthermia. So to repair that, what does the body do when there is hyperthermia? It does what? Vasodilation. And it does what else? Sweating. Who's the effector here? The sweat. The sweat is the effector. The sweat gland who secreted the sweat. And the blood vessels, don't forget the blood vessels, they dilate. So everyone who mediates the effect is an effector. What about the opposite? What if you get too cold? Now temperature went down. Is that hyperthermia or hypothermia? Hypothermia. How do we repair it? By how many effectors? How many effectors? By how many effectors? 2, right? So the first effector is vasoconstriction, and then shivering, thermal. What did I say earlier? Shivering thermogenesis. Very good, very good. And then we temperature goes right back up. Here temperature went back down. So here sweating and vasodilation brought temperature down. Sweating, you see sweating and vasodilation brought temperature down. And here vasoconstriction and shivering brought temperature up. After it was down, they brought it up. And here after temperature was up, they brought it down to the set point, euthermia. You see this now? Homeostasis, continuing. So homeostasis of blood pressure. We talked about that. The baroreceptor reflex. In homeostasis, in the baroreflex, what happens is when we rise from bed, the blood drains from the head and the blood pressure falls in this region. The blood pressure falls in this region. And that is detected by baroreceptors that transmit signals to the cardiac center of the brainstem. So here, who's the sensor? When the pressure went down, what was the stimulus? What was the signal? Hypotension, because the blood pressure went down. So that's hypotension. That's the signal. That's the signal. Who's the sensor? Who sends the signal? The baroreceptor. Excellent. Then the baroreceptor send a signal to the integrator. Who's the integrator? Who's the integrator who received the input signal, the input sensory signal that there is hypotension? Who received that? Yeah, the brain stem, the cardiac center of the brain stem. Then the cardiac center of the brainstem transmits signals to the heart to increase heart rate. Who is the effector now? The heart. The heart increases the heart rate, raising blood pressure and restoring homeostasis. So the barrel reflex illustrates the common components of a feedback loop. So we have a receptor. The receptor is a structure that senses. So here the sensor is the receptor that change in the body. For example, the baroreceptors above heart there are baroreceptors above the heart that monitor the blood pressure in the vessels. There is integrating control center, and that control center that processes the sensory information and makes a decision. It makes a decision and directs the response, for example, the cardiac center of the brainstem. Then we have an effector, an effector. This effector, okay, the cell or organ that carries out the final corrective action to restore homeostasis, to restore homeostasis. So you guys must always be able to develop your intuition. What is the stimulus? Hypo or hyper? Who detected the stimulus? The sensor. The sensor mediated input sensory signal to control integrating center, who will do negative feedback by sending output motor response. The output motor signal is going to go to the effector. The effector is going to restore homeostasis. Then we're going to terminate and stop. You must always be thinking about that until you get all of them. You're going to learn all of the negative feedback loops. Then you're going to learn what medications help those feedback loops. So don't just forget or just look at it and just like think about it like it's something. No, this is something that you're always going to be training your mind to grasp. Okay? So now let's look at here. A person rises from bed. So the blood drains from the upper body, creating homeostatic imbalance. So now there is an imbalance. There is less blood in the head and neck compared to the rest of the body. So now the baroreceptors, they sense that stimulus, and the baroreceptors above the heart respond to the drop in the blood pressure. And then there is an integrating control center where the baroreceptors send signals to the cardiac center of the brainstem. Then the heartbeat accelerates in response to the cardiac center. Then the blood pressure rises to normal and homeostasis is restored. Positive feedback and rapid change. So positive feedback is a self-amplifying cycle. Positive feedback is a self-amplifying cycle, and it leads to greater change in the same direction. We talked about positive feedback. We talked about positive feedback. It's a self-amplifying cycle, and it leads to greater change in the same direction, as opposed to the corrective action of negative feedback. The normal way of producing rapid changes. For example, we talked about that. Childbirth. When the baby is coming out, the more the baby stretches the cervix, the more the uterus pushes, expel the baby out. Blood clotting. When we are injured, we form a platelet plug, forming a thrombus, plate plug formation, and there is protein digestion. And then the more platelets attach, the more extra platelets attach. There's protein digestion and there's generation of nerve signals. It can sometimes be dangerous. So there's a vicious circle of runaway fever. Okay, this is positive feedback in children. Positive feedback in children. So what happens is brain stimulates the pituitary gland to secrete. So what happens? What's happening? Number one, the head of the fetus pushes against the cervix. So there's a stretch. Who's the sensor? Who's the sensor here? Mechanoreceptors in the cervix. So the sensors are always either mechanoreceptors, stretch receptors, or thermal receptors, or chemoreceptors, or nociceptors. So nerve impulses. Nerve impulses from the cervix are transmitted to the brain, and the brain stimulates the pituitary gland to secrete oxytocin, and then oxytocin stimulates the uterine contractions and pushes the fetus toward the cervix. So now do you see that this is a positive feedback? Who is the integrator here? Who is the integrator in this loop? I'm sorry? Who's the integrator? I can't hear you. Who's the integrator? Nerve impulse? The hypothalamus, the brain. How are the nerve impulses an integrator? Integrator means you are a computer. You have to be an organ. You have to be a hypothalamus. You have to be the pancreas. But nerve impulses are the input. They are impulses. They are being carried. They are information. They are telling me there is hyper pushing. There is mechanical sensation. So they're not the integrator. Who is the integrator? Yeah, so here they said it's the brain, but it's deeply is the hypothalamus. Very good. And now, what is the output signal? The output signal, the correcting output signal, what is it? The output signal goes to the effector. So who's the effector? Who is the effector who did the job? I'm sorry? Oxytocin is an effector. The effector is an organ. The effector was sweat gland. The effector was blood vessels. The effector was the skeletal muscles and shivering. So oxytocin cannot be an effector. The effector has to be an organ. The integrator has to be an organ. So who is the effector? The baby is not an effector. The uterus. The uterus is the one who pushes. So what is oxytocin doing? Oxytocin is telling the uterus, push. So the uterus is the effector. Who told the uterus to push? The hypothalamus. That's the integrator. How did the hypothalamus tell the uterus to push? Through oxytocin. So that's the output signal. So who is the input signal? The stretch receptor sensation. So the receptor senses push, it will send electrical sensory stimulation. Who integrates that? The hypothalamus. Who is the output signal? Oxytocin. Who is the effector? The uterus, pushing the baby out. So you must always be thinking, identifying all these guys. Now we're going to move into gradients. We talked about that. We said that matter and energy tend to flow down gradients. So a gradient is a difference in the chemical concentration, in the charge, in the temperature, or pressure. So I didn't talk about pressure. I talked about chemical concentration. If there is 500 glucose, 100 glucose, they're going to even out at 300 glucose on both sides. If there's 500 temperature degrees Celsius, 100 degrees Celsius, both are going to even out to 300. If there is 500 millivolt, 500 sodium, and there is 100 sodium, they're going to even out at 300. The only one I forgot was pressure. So here in pressure, we move from an area of a high pressure to an area of a low pressure. So when we're breathing, when you take a deep breath, all the air goes inside the lung. The lung has high partial pressure of oxygen. But the tissue, when you're running the marathon, it has no oxygen. So it has a low partial pressure of oxygen. So now oxygen is gonna flow from where? From your lung that has high oxygen pressure to your skeletal muscles that are burning out all the oxygen in the marathon, because there is so much aerobic respiration, there is so much oxidative phosphorylation consuming so much oxygen. So now oxygen moves from the lung into the skeletal muscles. So now do you see the gradient? Chemicals flow down a concentration gradient. So charged particles flow down an electrical gradient. And electrochemical gradient is a combination of concentration electrical gradients. So the heat flows down a thermal gradient. So the movement in the opposite direction is up the gradient and requires spending metabolic energy. So now moving down gradient, that shows going from 500 to 100. But we will learn in this course that there are some very important enzymes like the sodium potassium ATPase. It works the other way around. It takes from 100, pushing it back out. So if you open this classroom door, there's 500 people outside trying to get in. 3 people come in, then the rest will follow in. That's down gradient from 500 outside to the inside of the room. But imagine you had somebody here who was able to open the door and take out the 3 people who walked in or push them back against the 500. Now, there has to be a really strong person to do that. So we have enzymes that do just that. When sodium walk in a little bit inside the cell, they drop it. They take it from inside, throw it back out. And that is known as the sodium potassium ATPase. It will take sodium from in, where there is a little bit, throw it back out where there is a lot. And take potassium from outside, where there is a little bit, throw it back in where there is a lot. And for that, it uses ATP. That's why they call it sodium potassium ATP. It must get paid to do this really difficult job. Okay, flow down concentration gradient. So that shows you high pressure. So if you are a blood vessel like the aorta and you are so close to the heart, and the heart is a pump, it just pump so hard. Do you think the pressure in the aorta is going to be high or low? It's going to be very high. But the pressure very far near your leg, is that going to be high or low? That's why the pressure goes from your heart all the way to your legs. Then we have dietary glucose. That shows you so this is pressure gradient. What gradient is this? When you have a lot of glucose outside, green, and a little bit flowing in. So see the arrow? The arrow shows you that there's a movement of dietary glucose from outside to inside. Okay? So that's an example of what? Chemical gradient, concentration gradient. Then what is this? Where you have positive, a lot of positive outside, a little bit of positive inside. So the movement is going to be from where there is a lot of positive to where there is a little bit of positive. And the opposite, there is a lot of negative inside. So the movement is going to be from inside to the outside. That's an example of what kind of gradient? Electrical gradient. Excellent. What about heat? So you have really hot blood, and then all the heat is moved out. So the heat flow down thermal gradient to the cool air and the sweat. That's an example of what kind of gradient? Thermal gradient. Okay, so now the last part is we're going to talk about the language of medicine. So about 90% of our current medical terms come from 1200 Greek and Latin roots, reflecting the ancient past and the contributions of the Greeks and Latin into our medicine. Okay, so the Renaissance brought progress, but also confusion. So some structures named differently in varied countries, and some structures named after people. So in 1895, anatomists established worldwide naming conventions. They rejected eponyms, so used unique Latin names. So the Terminologia Anatomica provides standard international anatomical terms, provided Latin names and English equivalents. In 1998, it was approved by anatomists in over 50 countries. Analyzing the medical terms. So anatomical terminology is based on word elements such as the roots, prefixes, and suffixes. So the scientific terms. So one root or stem with a core meaning. Combining vowels, join roots into a word, prefix and suffix may modify meaning of root word. So the acronym. So the acronym is pronounceable words formed from the first letter or first few letters of a series of words, like a PET scan, positron emission tomography. So that's an acronym. So the plural forms of anatomical terms vary. So the cortex, cortices, the corpus, corpora. So the adjective often follows the noun it modifies. So the biceps, brachii. So the adjectival form of a term can appear different than a noun form. So for example, you have brachium and referring to the arm versus brachii, the adjective referring to of the arm. Okay, pronunciation. So simple pronunciation guides for many terms are given in the text when terms are first introduced. So pronunciation guides are also available online. Okay, so be precise in the use and spelling of terms. So many terms are spelled similarly but have very different meanings. So healthcare professions demand precision in order to maintain patient safety. So the key unifying principles of anatomy and physiology. So the unity of form and function. Anatomy and physiology complement each other and cannot be divorced from one another. Cell theory. All structure and function result from the activity of cells. Evolution. The human body is a product of evolution. Hierarchy of complexity. The human structure can be viewed as a series of levels of complexity. Homeostasis. Homeostasis, the purpose of most normal physiology is to maintain stable conditions within the body. Gradients and flow. Matter and energy tend to flow down gradients. The radiography, X-rays. So over half of all medical imaging is done by the X-rays. So they penetrate the tissue to darken the photographic film beneath the body. Dense tissue appears to be white. The radio-opaque substances can be injected or swallowed to fill the hollow structures. For example, blood vessels, the intestinal tract. There's a digital subtraction angiography, DSA. It is useful for showing the blockages and blood flow. This is a radiologic image of the head, X-ray, the radiograph, and this is radiologic images of the head, the digital subtraction angiogram. Okay, medical imaging. Computed tomography, CT scan, formerly called a CAT scan. So there's low intensity X-rays and computer analysis. So CAT scan. Okay, there's also, it increases the sharpness of the image. And then there is the MRI, magnetic resonance imaging. It is superior quality to the CT scan and no X-ray exposure. It is best for soft tissue. So it is best for the soft tissue, the MRI. And then functional MRI shows the real-time changes in the brain. Radiological images of the head, computed tomography. Magnetic resonance imaging, MRI. Medical imaging. So positron emission tomography assesses the metabolic state of tissue. It injects radioactively labeled glucose, and the image color shows the tissues using the most glucose at the moment, and damaged tissues appear dark. So the PET, the PET scan, positron emission tomography, assess the metabolic rate of the tissue, and they inject radioactively labeled glucose. Radioactive images, radiological images of the head show positron emission tomographic scan. That's a PET scan. Then medical imaging, sonography is the second oldest and second most widely used. It is high frequency sound waves that echo back from internal organs, and it avoids harmful X-rays. So good for obstetrics. The images are not very sharp. So that shows fetal sonography. So the face of the baby. Okay, thank you very much. Um,